So let me just sort of first thank you very much for agreeing to speak to us. This is a joint seminar series between CA2DM and the Material Science and Engineering Department. And when you were appointed as the new director to the Cancer Science Institute about a year ago, I think Antonio had the idea that these research centers of excellence should be talking to each other and exchanging ideas and knowing what's happening, between the different centers. And hopefully maybe there might be collaborations and exchange of information where we can learn from each other. And so in that sense, we're really delighted that the director of the newest Center of Excellence is one of the panelists, Kostya Novoselov, as well as Vinicia Rosa, who's in the area of biomaterials. The structure of these seminars is that usually it would be no more than 40 minutes of a formal presentation, so keep it short. We'll start off the discussion with the panelists. And the idea there is that to make the seminar a bit unique, there was all these online seminars. You can just watch these on YouTube videos, and this is to sort of localize it a little bit, and so have a little bit of local interaction. So the panelists will engage you in some discussion, and then we'll slowly open it up to the rest of the audience for question and answer, and other people can sort of join the discussion. So that's the plan. So without much ado, let me introduce you. So as I already mentioned, Professor Ashok Venkitaraman is not so new now, he's been here a year, but about almost exactly a year as the director of the Cancer Science Institute. And he's credited for discovering how do you pronounce it, BRCA gene, BRCA gene? Actually, on a very personal note, I mean, a member of my family got breast cancer about 2 years ago, and one of the first tests they did was to test for this gene. And apparently, if I remember correctly, the having this or not having this gives you like a 5 to 10-fold more risk of getting variants that are not treatable by drugs. And so your entire treatment is determined by your discovery. And so I think you've already had a huge impact on society and the world. And we welcome you telling us about that story and other things that you're working on currently.
Thank you very much for the introduction, Shafiq. It's a great pleasure to be speaking here this morning. I should say at the outset, I'm neither a material scientist nor a physicist, but I hope that I can give you some insight into some of the methods and approaches that we've used to look at the breast cancer protein, BRCA2, inside living cells and try to understand how it executes its structure and function. So what I'm going to do is very briefly, in about 30 to 40 minutes, share with you some vignettes of the last several years of work in my laboratory on how we've approached analyzing the structure and function and transport of the breast cancer protein, to understand how mutations affecting this protein can actually cause cancer. And what I'd like to end with is perhaps some thoughts on what does this mean in terms of general implications for the way that we approach the prediction and analysis of protein structure and function, particularly in the case of disease-causing proteins like BRCA2. So let me begin with some fundamental premises that will underlie my talk, which I suspect many of you will be familiar with. Cancer as a disease is, in my opinion, unique amongst human diseases. We know that changes in the genome, in the DNA, can predispose people to disease. But in the case of cancer, changes in DNA, mutations, in other words, actually cause the disease. In other words, as you progress from normal tissue on the left-hand side to carcinoma on the right-hand side, the cells within these tissues, which are first normal, acquire faults in their genome, in other words, mutations. So the instability of DNA, which is normally quite stable in normal cells, is an almost universal feature of what makes normal cells cancer cells. And these mutations essentially confer changes in protein structure and function that enable cancer cells to behave badly, as I call it, in other words, to escape the normal rules that govern cell behavior. You might be thinking, if this is the case, how does one explain the fact that we have known for many years that the origins of cancer are not purely genetic? In other words, environmental influences ranging from soot in chimneys, which was first identified in Victorian times to cause skin cancer, all the way through to the well-established connection between smoking and lung cancer. How do they work? And the answer is we don't fully know, but we suspect that there is a collusion between certain environmental influences and the way that genes are mutated and or regulated in our cells, which can enhance cancer risk. So in this background, my lab has for the past couple of decades been trying to answer two relatively simple yet obviously complex questions. What is it that makes certain people more susceptible to cancer than others? And as a background to this, I would say that we know that most cancers occur in older people and that the majority of cancers will occur in only a fraction of the population. Some of this is genetics, some of this is environment, but we don't know how this interplay works. The second question that I'll briefly allude to towards the end of my talk is trying to use this understanding to develop new means to delay or even prevent the onset of cancer. And this is my aspiration, to convert the knowledge of fundamental mechanisms into clinical impact in this way. So let me begin by giving you a bit of background about how our genetic inheritance may influence cancer susceptibility. So here I'm sharing with you a report a few years ago of the so-called flagship project conducted at the National Institutes of Health, the Cancer Genome Atlas, or TCGA project. In this project, over 10,000 cancers from 33 different organs were analyzed at the level of their genome, and in some cases at the level of the RNA as well as the DNA. And if you had asked me before these results were published, I would have told you, I think that inherited mutations that confer cancer susceptibility will be relatively rare. But as you can see, this is not the case. And in fact, nearly 10% of cancers unselected, just seen in the clinic, the DNA of these people, not in the cancer cells, but in their normal cells, carry known pathogenic or likely pathogenic variants in many different genes that affect cancer susceptibility. And indeed, some of these are more frequent in certain tissues, for example, in breast cancer, ovarian cancer, pancreatic cancer. And mutations affecting a small group of genes, I've highlighted just the three that I happen to work on, but there are a few more groups. In this case, the breast cancer genes BRCA1 and BRCA2 and their partner PALB2 frequently recur. So I emphasize that these mutations are carried in the germline. In other words, these are not somatic mutations acquired in cancer cells, but rather inherited mutations from your parents that increase the risk of developing certain forms of cancer. So I hope you'll see that having 10% of all cancers being in some way related to these germline mutations is unexpectedly high. So how can we understand the structure and function of these proteins encoded by these susceptibility genes? And how can we predict from amongst the hundreds or thousands of variants that occur in the genome, which are most likely to confer cancer risk? And can we distinguish and differentiate that cancer risk? And to illustrate how we have approached this problem, I'm going to use as an example the breast cancer gene BRCA2 that we've been working on for about 20 years in my laboratory since it was discovered through reverse genetics. So I'm just going to introduce you very briefly to some background information. If you look at most forms of epithelial cancer, these are all the common kinds of cancer: colorectal, lung, breast, ovary, they're all arising in epithelial cells. The rule is that generally about 5 to 10% of these cancers are evidently occurring at a higher frequency within families. In other words, there is a genetic bias. And about 90 to 90% are so-called sporadic. In other words, there's no evident familial bias. Of course, please remember that 1 in 3 of us is expected to develop cancer at some point in our lives. So it is difficult to know whether even some of these sporadic cases do have a familial or genetic component simply because of the frequency. But if we look at a well-studied example, breast cancer is one of those, you'll see that of the familial cases, we understand for breast cancer roughly half of the genetic risk. Roughly half of the genetic risk. It's quite remarkable in the stage of DNA sequencing that still about half of the familial risk in the genes is unattributed and probably is accounted for by multiple genes which have a low effect or low penetrance genes, we call them. However, the genes that I've been working on, BRCA1 and BRCA2, the breast cancer genes, are high penetrance genes. In other words, if you inherit just one single mutant copy out of the 2 that you have in your genome, you have a very high risk of developing not just breast cancer in the case of BRCA2, but cancers in many different tissues. And you can see that this is a highly penetrant risk, and also that the cancers tend to occur much earlier than is normal. In other words, it's as if the process of carcinogenesis is accelerated somehow in these mutation carriers. So we have studied BRCA mutations as a very powerful model for trying to understand and enact early intervention in cancer. And we focused in our laboratory studies on trying to assess the collusion between genes and environment in these families, to understand in detail the molecular and cellular events that lead to carcinogenesis, and to develop enabling technologies that will enable us to convert these pieces of information into something we can use in the clinic. Our work has really focused on these main questions: which BRCA variants are pathogenic, why? What steps lead to BRCA mutant cancers? And of course, how can we develop technologies? Our goal in the long term is threefold. We want to be able to detect cancers arising in these mutation carriers at a very early stage. We want to be able to understand which of those cancers is a high risk, highly progressive, aggressive cancer and needs quick treatment. And we want to develop clinical interventions that are appropriate to the risk. So I'm going to speak for a little while, going down now to mechanistic level studies on some of the steps that lead to BRCA mutant cancers, and in particular, the role of BRCA2, which is the protein that we've studied in my lab. So again, by way of conceptual background, let me introduce you to a paradigm which was posited by Al Knudsen in the 1970s to explain inherited cancer risk. It's called the Twohit paradigm, and essentially it posits that in cases of cancer risk genes like BRCA2, you can carry a single copy mutation, which is pathogenic, but you have to lose the second normal copy in certain cells, and is this inactivation that actually leads to the loss of function of this gene and thereby leads to cancer. And this influenced our earlier studies because we thought BRCA2 was likely to work in this way because of the way that its mutations occur in families. And we created, in my Cambridge lab at the time, I was just starting my lab up at the MRC Laboratory of Molecular Biology, so we created a knockout model. And in that model, we looked and observed that chromosomes, which are the carriers of the DNA in cells, are grossly distorted, and there is an inability to maintain either the structural or the numerical integrity of these carriers of genetic information. In other words, BRCA2 inactivation promotes cancer, we believe, by crippling genome maintenance and causing genome instability. And indeed, we now know through the work of many different laboratories nearly 2 decades later that BRCA2 has critical functions in maintaining the stability of the human genome, particularly in enabling DNA copying or DNA replication to proceed normally and enabling the repair of lesions such as broken DNA, which can occur during replication. But more about this later. This is just to give you a perspective and a background. So with this by way of background, I'm going to dive deeper into the topic that I'd like to discuss with you as material scientists and physicists. How can we envision and analyze the structure, function, and transport of proteins like BRCA2 within the context of the intracellular environment? So here's the BRCA2 protein encoded by a very, very long gene. In fact, BRCA2 in humans is a massive protein of about 3,500 amino acid residues with an apparent molecular weight approximating 400 kilodaltons. Its structure is unresolved. It simply does not look like any other molecules that we know, so it's hard to infer function from homology. We do know that it works to form complexes with many different partner proteins, and these complexes are essential for it to execute its functions. Finally, we know that BRCA2 is made in the cytosol and is transported to the nucleus. This again is a non-trivial process because you will understand that transport into the nucleus of proteins that are not small enough to get there by simple diffusion, such as BRCA2, has to also be tightly regulated and controlled, and is in fact contingent on aspects of structure and folding. So within BRCA2, let me simply draw your attention to a couple of motifs that I'll allude to in my talk. Look here, and you will see these 8 red bars. These are very short, roughly 30 to 35 amino acid motifs. We find that they are conserved not only in their sequence, their amino acid sequence, but also in their spacing. And these 8 sequence motifs, called the BRC repeats, are in fact a feature of almost all mammalian forms of BRCA2, going all the way back to simpler organisms like rodents, or across 30 to 50 million years of evolution. And I emphasize that their sequence as well as their spacing within the middle of the BRCA2 protein is conserved. There's also another conserved domain here, more about which I will speak later. But this is a domain which appears to bind partner proteins and also fold into a structure that can engage, bind to DNA. And I'll say more about this later. So I guess before I start to tell you about some of the approaches that we've taken to analyze BRCA2 protein structure and function, it behooves me to make a few remarks, which I'm sure you're all more familiar with than I, regarding the intracellular environment and its complexity. So typical mammalian cells are very complex in their organization, as illustrated here. In particular, besides the nucleus and the cytosol enclosed by this lipid bilayer or plasma membrane, there are a number of membrane-bound organelles, example mitochondria, which are the energy-generating organelles that synthesize ATP, as well as the so-called endoplasmic reticulum and the Golgi apparatus, which are sites of protein synthesis and transport. If one looks by scanning cryoelectron tomography at, for example, this region of a cell, you begin to get a glimpse of the complexity and the crowding of membranous and non-membrane enclosed structures. So here you're seeing, for example, bounded in yellow, sections of the endoplasmic reticulum. In orange, the free ribosomes, which are the protein synthetic machinery, the green mitochondria, which are the energy generating organelles, and in red and purple, different forms of vesicles, which transport proteins and other solutes within and outside the cell. But you can see in the pseudo-colored image derived from this type of scanning electron tomography image, how complex is the organization of the intracellular environment. This complexity is also reflected even in areas of the cytosol. And this is an illustration of a bacterial cytosol, but much of what is true there is also true for human cells. And you see here, even in the cytosol, there are structured machines such as ribosomes and the protein translation apparatus shown in pink, a number of folded proteins which are compact, you can see, and importantly, a number of disordered proteins. This is a highly crowded environment with protein concentrations ranging from 50 to 250 grams per liter, and nucleic acid concentrations in the nucleus and cytosol approximating 20 to 50 grams per liter. In other words, macromolecules can occupy between 1/10 to nearly half of the total volume of cells, suggesting how crowded the environment is. So how does one begin to analyze the structure and function of proteins like BRCA2 in this environment? Let me share with you some studies performed some years ago in my lab, in which we have tried to use in living cells methods of biophysics, in this case, fluorescence correlation spectroscopy. And you know that this method works essentially statistically. We illuminate and measure fluorescence emissions from tagged fluorescent molecules in a small volume, which we can isolate within the cell nucleus. And we can observe fluctuations in molecules that diffuse through this volume or remain static within this volume. And from these measurements, infer aspects of their diffusion, size, mobility, etc., within living cells. Of course, one of the complications here is how do you introduce fluorescent tags into molecules in living cells? And in order to keep the system as natural as possible, we use genetic engineering, in which we knocked out one of the 2 copies of BRCA2 or partner molecules and tagged the other copy with a fluorescent tag endogenously within the genetic locus. So essentially, we're looking at fluorescent, but otherwise normal and functional molecules of BRCA2. And in these early experiments, several things became apparent, which illustrate the complexity of protein structure and function in this complex environment. So let me say that we observed that some molecules of BRCA2 were resident within the interrogation volume for a sufficiently long time that fluorescence photobleaching essentially made them invisible. So these really massive immobile complexes are excluded from the analyses I'm going to show you. But we can measure the fluctuation of diffusing particles of BRCA2 tagged with a green fluorophore, EGFP. And what I'm showing you here are histograms in which we have binned around diffusion constants, time of diffusion within this defined interrogation volume, and these bins are in milliseconds. I'm showing you here in the black bars the typical diffusion of a very small protein, about 30 kilodaltons, which is just free GFP, freely diffuses, doesn't bind to anything, and you can see what it moves like. And I hope you can see in the gray bars that BRCA2 EGFP has a very different behavior indeed. There are a couple of aspects that are remarkable. The first is, as you can see here, that there is a very slow-moving component of BRCA2. This is too massive in terms, if you apply simple Bose-Einstein diffusion kinetics to represent a molecule, rather we think it represents anomalous diffusion of unfolded BRCA2, simply bumping into other macromolecules which slow its diffusion in this crowded environment. And there is a faster diffusing component, which again, using diffusion from the diffusion constant, approximates to the molecular weight and molecular mobility of BRCA2 with a few associated partner proteins, more about which later. The second remarkable thing is what happens when we induce DNA damage in these cells. So you can see here the average percentage of BRCA2 in the fast-moving component here, and you'll see it's about half. And if we induce DNA damage, you can see that this rapidly increases within minutes. In other words, it's as if BRCA2 is shifting from the slow diffusing pool into a faster diffusing pool. We know that this is a result of changes induced by the DNA damage response, because if we inhibit the DNA damage response using this chemical inhibitor, wortmannin, we can also inhibit the changes in molecular diffusion. We can use fluorescence cross-correlation spectroscopy, again in living cells, and again a method that I'm sure you will be familiar with, to look not only at BRCA2 labeled with green EGFP, but one of its partner proteins, the DNA repair enzyme RAD51, and ask using cross-correlation whether the two form complexes and whether the complex formation is influenced by DNA damage. And indeed, as you can see here in these cross-correlation index plots, this is BRCA2 green plus just free cherry as a control. The two molecules have virtually no cross-correlation within the interrogation volume. And you see here that in contrast, BRCA2 labeled in green and cherry labeled RAD51 do have a cross-correlation index, from which we can estimate changes that occur before in gray and after in blue DNA damage induced by ionizing radiation. And I hope the conclusion is quite clear that damage not only induces the mobilization of BRCA2, but it also enhances the fraction of BRCA2 that interacts with a repair enzyme, which appears to be a key cargo that helps BRCA2 to execute its functions. So from this, you can see that we can apply methods combining genetics, somatic cell genetics, fluorescence microscopy, and simple statistical spectroscopy correlations to begin to develop models of how massive proteins like BRCA2 may exist, diffuse, and potentially fold. And our working hypothesis at this time is that BRCA2 in the slow-moving complex is probably largely unfolded, and the anomalous diffusion is inhibiting its mobility. There is a faster-moving component, probably partially folded, if not fully folded, which probably is interacting with partner proteins. And the DNA damage causes a mobilization from what we call a sequestered inactive pool into an active pool. And this does indeed correlate with the functional formation of a complex with an enzyme RAD51, which repairs DNA. So the conclusions that we came to from these early studies is that BRCA2 is not only a massive protein, but one that probably is largely, if not fully, intrinsically disordered. We and others have shown in the intervening years that regions of BRCA2 with known structure that has been resolved either by cryo-electron microscopy or by X-ray crystallography tend to exist in complex with other partner proteins. In other words, complex formation seems to confer order. And you can illustrate this in this sort of crude prediction using the very poor algorithms that we have today that can predict structure that a lot of BRCA2 is likely to be intrinsically disordered, the red regions, the blue regions representing positions of potential order. And you can see here some of the crystallographic structures that have been solved. For example, this was the first from my own laboratory in Cambridge, the structure of a complex between one of these BRC repeats and the cargo protein RAD51, which is clearly highly ordered. This solved by Nikolai Pavletich's lab at the Rockefeller, which is the structure of a complex between this region of BRCA2, a small partner protein called BSS1, forming a DNA binding structure. And more recently, this from Lawrence Pearl's lab in Sussex in the UK, again, the structure, a beta propeller of a partner protein, a fragment of a partner protein interacting with a region of BRCA2. And this illustrates some of the lessons, if you will, or insights that I'd like to convey to you. So let me now draw your attention to this really critical function of BRCA2, which is in the repair of DNA, particularly during the copying of DNA. So what you can see here is a process of repair of DNA. And you can see that when DNA is broken, it can be repaired either by a simple process of sticking back the ends, which is, as you will imagine, a very error-prone mutagenic process. So it's not a preferred process in cells. And here, an error-free process in which a broken copy of DNA can exchange genetic information with an intact sister copy on a homologous chromosome or in a replicated copied sister chromatid, and thereby effect a relatively error-free form of repair. And this process is called DNA recombination. We discovered in my lab many years ago that BRCA2 is essential for DNA recombination. And we also know that it works to transport the enzyme that I referred to a few minutes ago, RAD51, a small 37 kilodalton protein, to the sites of DNA damage, where RAD51 forms ordered assemblies on DNA, which actually mediate the reactions that lead to the exchange of genetic information and repair. So let me tell you a little bit about our work in this area and how it informs our knowledge of the structure and function of BRCA2. So this was the crystal structure that we first solved in Cambridge in collaboration with Luca Pellegrini and Tom Blundell. And this shows you in blue and magenta the atomic structure. This is the backbone, the alpha carbon backbone shown in a ribbon representation. In blue and magenta, the core catalytic domain of the enzyme RAD51, and in green, the structure of the bound BRC repeat, which I remind you is one of these 8 30 to 35 amino acid motifs. So what you can see here is that the core catalytic domain of RAD51 at its center has this somewhat planar structure formed by these anti-parallel beta strands. And you can see that the interaction with BRC, repeat BRC4, extends the structure by an extra 2 anti-parallel strands. And you can also see the spring-like 3,5 beta hairpin, which is held together by intra-chain hydrogen bonding, which opposes this piece of BRCA2 against this piece of RAD51. And we realized when we looked at that sequence in greater detail, quite remarkably, not only is that sequence represented here with the particular conservation of the phenylalanine and alanine residues, not only is it conserved in all mammalian BRC repeats, it's actually present in RAD51 itself in a region which is disordered in the structure, but is positioned towards the N-terminus of RAD51 extending in this region. In other words, the target protein RAD51 itself has a copy of the BRC repeat. So it's as if BRCA2, a molecule which comes up later in evolution than RAD51, which is quite ancient, has copied or mimicked this motif. And this insight led us to begin to question how the BRC repeats might regulate the process of DNA repair by RAD51. So it took us 10 years of work to reconstitute in a test tube the reaction of recombination using recombinant proteins, RAD51, pieces of BRCA2, such as the 1,000 amino acid segment containing all of the 8 BRC repeats, and single strand and double strand DNA substrates. We first had to overcome some skepticism by biochemists who were working in this area for many years, because they said, you know what, we can take RAD51, stick it together with single strand and double strand DNA, and we get this recombination reaction to work just fine. Why do you need BRCA2? And what Mahmoud Shivji, a biochemist working in my lab, discovered was that yes, it does work fine, but only if you have 1 to 3 millimolar calcium or 100 millimolar ammonium ions, which I hope you will agree are probably not typical of the intracellular environment. But in physiological ionic conditions, what Mahmoud found was that there is, if you will, a premature engagement of RAD51 with the double-stranded DNA when it should be binding to the single strand first in order to order this reaction correctly. And only in the presence of BRCA2 is the order of things reversed. BRCA2 BRC repeats, we found biochemically, stabilize the single strand interaction, inhibit the double strand interaction, so the reaction can now proceed in order through these opposing activities of the small segment of BRCA2. How does this work at the cellular level? To address this, we've used super-resolution microscopy methods, particularly DStorm, which I'm sure that many of you will be familiar with. It's a method of single molecule detection, which essentially relies on the blinking of standard organic fluorophores, which we can use to label particular biomolecules. These fluorophores can be induced to blink using the appropriate chemical conditions, and we can capture thousands of images of these blinking fluorophores and reconstruct single molecule resolution from integration of these images. And you can see the dramatic effect here. Here is RAD51 in pink, assembling at sites of DNA damage within a cell. Here's our typical confocal image at the diffraction limit. And here is a super resolution image constructed by DStorm. We can actually begin to see structures of RAD51 inside these cells. And indeed, what we see, and we can reassemble this using both scatter plots, which are depicted here, or indeed Voronoi representations, polygon representations, which give us a localization precision in the 2 to 10 nanometer range, so quite high resolution in these terms. And we can begin to actually analyze what happens in the reactions that lead to recombination mediated by BRCA2. I'm just going to show you a few of these images. These are in standard laboratory cells, the HeLa cell line, exposed to ionizing radiation, and we've taken a time series and we've stained either for a molecule called replication protein A, which binds to single-stranded DNA, which we can also label. And you can see here the assemblies of RPA on single-stranded DNA shown in magenta and green proceeding over time. And you can see the co-localization of these proteins over time after we induce DNA damage. But now it begins to get interesting. What about RAD51? And here I hope you can see beautifully how over time RAD51 in magenta assembles, co-localizes with single-stranded DNA labeled in green by replication protein A, and how these filaments of RAD51 grow over time until they extend to hundreds of nanometers in length within a cell. And we know that these reactions depend on BRCA2 because if you remove BRCA2 by genetic means, it stops here. Rad51 does not even localize correctly to the sites of DNA damage, let alone form filaments. We can also do more subtle experiments in which we delete, for example, that C-terminal region of BRCA2, which we also know to bind Rad51. And we get this very intriguing result, which is that Rad51 does localize to sites of DNA damage marked by BRCA2, but it does not grow into filaments. This is the normal cell, and this is an isogenic cell, except that it lacks just this segment of BRCA2. So I hope you can quite clearly see how Rad51 gets there, but somehow its assembly into a long filament doesn't proceed without this region of BRCA2. So from this type of study, we can actually begin to assemble a picture of how the different regions of BRCA2 serve to bring Rad51 to sites of DNA damage and to stabilize the elongation of these long filaments formed by Rad51 at these damaged sites. We have begun to explore these at near atomic resolution using cryoelectron microscopy. And here's some work we published a few years ago in which we have used cryoelectron microscopy to resolve at between 3 to 4.5 angstroms the structure of Rad51 filaments on single-stranded DNA, the so-called pre-synaptic filament. And you can see here 3 monomers of Rad51 assembled in a helical filament around this core of single-stranded DNA, seen here in a coronal section with the DNA right in the middle. Beautiful helical conformation. Formation of this, as I said, actually somehow depends on reactions catalyzed by the BRC repeats of BRCA2. So we have taken our crystal structure and begun to ask how the BRC repeats might map onto the Rad51 multimer. And you can see here that this type of superposition reveals at least 2 interactions. One, the inhibitory interaction that we predicted nearly a decade ago, where this 3,5 beta hairpin structure repeated in Rad51, you can see that in yellow and in red in BRCA2 superimposed, actually forms the protomer-protomer interface, just as we predicted from the monomeric structure. And a second interaction, which does not disrupt the protomer-protomer interface, but rather may serve to help to stabilize the growth of this filament by binding, if you will, to the side of these monomers as they assemble on DNA. So this has enabled us to get some insight, structurally speaking, about how BRC repeats may stabilize the forms that we recognize biochemically, the single-stranded filament and the extended double-stranded filament, using interaction with distinct motifs. But I'm not going to go into this in great detail. I'd be happy to discuss it later if questions arise. Instead, I'm just going to share with you in the last few minutes some ongoing work where, again, we have tried to ask, how does the BRCA2 BRC repeat structure influence the assembly of Rad51 on double-stranded or single-stranded DNA substrates? And here I'm showing you our recently resolved by cryo-EM double-stranded Rad51 filaments and single-stranded Rad51 filaments, very similar looking helices, except of course that the helical parameters are different in that the double-strand filament is more compact, as you would imagine, because the core is thicker. It's got 2 strands of DNA, and the somewhat more extended single-strand filament around a thinner core of single-strand. And here's a new structure, which is one that forms in the presence of a few of these BRC repeats, genetically engineered so as not to inhibit filament formation. And you can see that this does form an intermediate structure. But what is remarkable and very exciting to us is that there is a new incoming strand of DNA, which we didn't put there. we form this in the presence of BRCA repeats, in the presence of just a single-stranded oligo-DT DNA. But what you can see is only in the presence of the BRCA repeats, you now have an incoming identical oligo-DT strand, which of course cannot form complementary Watson-Crick base pairs, but instead is catalyzed to form aberrant wobble base pairs, which are non-canonical and therefore hold the sugar phosphate backbones at a distance, but nevertheless, we're beginning to get a glimpse of how the BRCA repeats may enable the formation of homologous pairs between single-stranded DNA and incoming duplex DNA, which has been melted or unwound. So these are studies in progress, but I hope illustrate to you how deeper structural biology can actually give us quite deep insight into the structural transitions that enable the reactions catalyzed by BRCA2 in living cells. So I'm going to end by asking a simple question. What does this all mean in the context of BRCA2? Why should we begin to understand and elucidate aspects of protein structure, folding, and function? And the simple example, I hope, will convince you why this is important. And that is the fact that truncations affecting BRCA2 account for about 70% of all of the pathogenic mutations. But about 30% of the mutations in BRCA2 that are associated with cancer substitute with a missense codon a single residue in this massive, massive protein. And we don't understand if or how these missense mutations may cause cancer. So over the last couple of years, we have actually been looking at one particular cluster of missense mutations associated with cancer, which fall in this region, which was structurally characterized by Nikolai Pavletich's lab some years ago. It's a complex between BRCA2 and a small partner protein called DSS1, just 70 amino acids. And the structure that is formed has the following components: a helical domain, 3 copies of a so-called oligonucleotide oligosaccharide binding fold, which just binds to single-stranded DNA, and here a tall tower domain, which contains a double-stranded DNA binding motif. This region accounts for partner protein binding. This region accounts for DNA binding. And you will see most of the mutations in patients fall within these 2 domains. Through a series of biophysical, cellular, biological, and structural analyses, we have found very recently a model for how these mutations work to cause cancer. What we find is that this region of BRCA2, the DBD, has to engage with DSS1 in order to fold correctly, and that this correct folding is actually essential for the transport of BRCA2 to its site of action in the nucleus. These mutations in patients all, to varying degrees, inhibit or even completely obliterate the ability to bind to DSS1. In turn, this results in a lack of protein folding and assembly. These molecules form unfolded oligomers that aggregate within the cytosol of cells and therefore are non-functional because they're not transported to the nucleus. Excitingly, we find that some of these mutants in patients can still bind DSS1. So if we supply a small region of DSS1 in excess, we can shift the equilibrium towards a folded functional state, potentially revealing a therapeutic approach in these patients. So I hope this illustrates to you very clearly some of the issues that we have been contending with and why it is so important to address and answer these questions. How do we predict the folding and structure of proteins involved in disease when no structural information is available? or when they may contain some or vast regions that are intrinsically disordered. How do we factor in to folding and function the effects of the crowded, complex, membrane-bounded intracellular environment? Can we, using all of this information, begin to predict if someone has a mutation in a potentially pathogenic protein, whether that mutation is pathogenic or not, and how it works? These are some of the questions where I hope that our studies on this model system have elicited information that may help to persuade you and your colleagues that these are good model systems which we can work on together to answer these very fundamental questions about protein structure, function, and transport in the complex intracellular environment. Thanks for your attention.
Thank you so much for that wonderful talk. It's been a long time since I've formally studied biology, so a lot of it was very difficult, but at least I think I understood parts of it, at least that in order to solve the intractable problem of cancer, we need to first solve the intractable problem of protein folding.
I think that's a fair summary, Shafiq.
Anyway, let the experts comment. Kostya, Nithias, do you have any discussions for the?
I'm really curious that you're saying that most mutations leads to the protein folding rather than unfolding. So is it a general statement? Because mutations can happen anywhere. I would presume that some of them might promote folding, but some of them should actually do exactly the opposite. So what's the general knowledge on it?
Kostya, that's an interesting question. For many of the disordered proteins, for example, BRCA2, for example, the very famous tumor suppressive protein P53, these proteins seem to exist, as our results and the work of many labs illustrate, in a state of intrinsic disorder within cells, and they acquire order, structure, folding by interacting with partner proteins when their function is required. So we think that mutations in BRCA2, for example, prevent that ordering, in other words, the transition from disorder to fold. And in fact, the mutation cluster that we have analyzed, that I shared with you in my results, prevents the interaction of BRCA2 with BSS1 and leads to an unfolded state, which is non-functional. It aggregates in the cytosol. Other mutations in P53 are known to do the same thing. So I would say that for disordered proteins of this kind, mutations tend to change towards disorder rather than towards order, often by preventing interaction.
What do you call disorder and order? How do you define disorder and order in this case?
It's a very difficult question. We have an operational definition, which I illustrated to you at the beginning of my talk, which is that we think that in its unfolded state, BRCA2 undergoes really evident anomalous diffusion and is in a very slow moving state as measured, for example, by fluorescence correlation spectroscopy. In its folded state, when it binds to partner proteins like RAD51, we believe that that operational definition can be visualized and measured semi-quantitatively by transition to a more dynamic folded state in which we see diffusion consistent with the expected molecular mass of BRCA2 plus its associated proteins.
And do you see that the diffusion outside of the nucleus is in the folded state and then it unfolds inside of the nucleus, or any characteristics of that?
So our studies suggest that in order to get into the nucleus in the first place, it has to enter at least a partially folded state. But once in the nucleus, which, as you know, is extremely crowded in terms of macromolecular environment, much of BRCA2 seems to relax or unfold. And therefore, we think, as illustrated by our studies on the pathogenic mutant, that there are at least two steps in the maturation and function of this protein. One, a partial or some type of folding required for nuclear transport. And then within the nucleus, a sort of sequestration or a resting state, which may be more unfolded, which, when the protein is required after DNA damage, for example, transits in the nucleus to a faster moving, more folded state. So it's very complex. And because of the spatial resolution of FCS being too large, I mean, it's not resolved enough, we can't clearly distinguish some of these transitions in living cells.
And for me, it won't be a surprise that in the crowded environment, you get a little bit more unfolded state, because you start to interact with many other proteins outside, not only with itself, but then it probably it complicates the functioning of the protein as well. Do you understand the balance of the interaction here? So
no, we don't. I think your point is very well taken, and that's exactly what we believe, that in the crowded nuclear environment, there is a relaxed state, an unfolded state, because of the types of interactions induced by macromolecular crowding that you're talking about. But somehow, after activation of the protein, for example, by signals elicited by damage, we know that it binds to certain partners. Our cross-correlation spectroscopy experiments show that the folded, more dynamic mobile state is accompanied by an increase in cross-correlation with at least one binding partner, RAD51. And so we believe that there is an induced folding. And we think, we don't know what those signals are, but they could be post-translational modifications, for example, protein phosphorylation, which lead to interactions that then lead to folding. And we're trying to characterize some of that using mass spectrometry and proteomics.
So let me just add that so Antonio, who was supposed to be the host of this today's meeting, unfortunately couldn't be here, but he used to like to end these meetings on time. And since we started 5 minutes late, that gives us 5 more minutes of Q&A. And that's so people who are in the audience can start using the Q&A button to sort of ask questions. I'm sure there are several, but while we're waiting for people to ask questions, maybe Vinicius, do you have any comments or thoughts?
So hi, Prof. Thank you very much for this great lecture. And like Prof Shafik has said, being like in the same screen with the person from the BRCA gene is kind of very exciting. I have more of a curiosity. If we take other mammals, large mammals, right? The number of suppressing genes they have is like 10, 15, 20 times higher than ours. Is that by pure chance? Because people put everything on the count of evolution in a way, but evolution is totally random or whatever. Is there any sort of technology or future application where we could potentially or even benefit from increasing the number of suppressor genes on us, or we are?
That's a great question, Vinicius. So let me just give the audience a bit of background. So what Vinicius is alluding to is what is called Ketos paradox. All mammals start from a single cell. That cell has to divide, and in large mammals, elephants, blue whales, the number of divisions is so much more than in humans because our cells are all roughly the same size. So why don't these large animals, we know for elephants kept in zoos at least, get cancer much more often because the cells divide and acquire mutations much more often? And the answer, as Vinicius said, is that some of these mammals, like elephants, have multiple copies of these tumor suppressor genes, whereas we have 2 copies of p53. Elephants can have up to 30 copies of p53. So in fact, we are at the moment trying to analyze in the general population whether cancer susceptibility may correlate, not with copies, obviously, because we inherit a diploid genome, but with the level of expression of some of these proteins. And it would be really exciting because I think comparative evolution is teaching us that we might find ways to delay or prevent the onset of cancer if we can find ways to enhance the expression, folding, and function of tumor suppressors like BRCA2 or P53 in humans. So that is a topic that we are very interested in, and we're starting to analyze at the population level as well as at the laboratory level.
So while we're still waiting for questions from the audience, maybe I ask Kostya a question. Given the talk, what can functional materials help with solving these questions?
Yes, I would like to see if there is room for collaboration on an issue that Kostya and Antonio had earlier said that there is interest in your group, which is how do we predict the structure of macromolecules like proteins? How do we predict the nature of macromolecular interactions and how they contribute to protein function? There have been great advances in this area, for example, DeepMind and AlphaFold, but I hope you will all agree that there's a lot of work still to be done. And some of it requires the acquisition of experimental data against which some of the analytic tools can be tested. So my purpose in sharing with you some of the work that we have done on proteins like BRCA2 is to persuade you that here are some systems where we have the biophysical tools, the genetic tools, some knowledge of structure and function that could be used to gather data to develop and refine analytic techniques for protein structure prediction. Why is this important? I don't want to have to solve a crystal structure in order to understand whether a patient has a pathogenic variant in a protein or not. I don't want to have to solve a crystal structure in order to be able to develop potential therapeutics targeting these abnormal proteins.
Kostya, do you have any thoughts about that?
Well, I think that what is of interest for me is the problem of complexity. So clearly, we have an extremely complex system which finds itself a way through, and what we don't understand at all now is how to design such systems. And for us, it would be really a huge help to figure out the basic principles, how those proteins know which form to fold into and unfold into depending on external stimuli. And so we believe that we can build some very basic model structures which interact under similar interactions pathways and maybe exhibit some of the basic behavior which we find in the proteins. So, but we need to start somewhere, and just understanding the basic behavior of the proteins would help here.
That would be terrific, Kostya.
Since nobody in the audience asked a naive question, I'll take the liberty to ask the last and a very, very naive question. So if I understand correctly, this double hit model tells you that something happens to these BRCA molecules that cause them to slow down in diffusion or unfold or fold, that then impairs the repair mechanism within a cell. But it seems to me that if that one single cell, something goes wrong, that's less of a worry than how that cell affects the other cells. It must have some kind of signaling mechanism to other cells that's independent of this particular folding or unfolding. And so just wondering, isn't the signaling mechanism the one we want to understand as opposed to how a single failure mode of a single cell?
Shafiq, the situation is that people inherit one mutant copy of BRCA2, but cancers often contain 2 mutant copies. So how do you get that second inactivating lesion? Interestingly, we've recently found, and I didn't have time to talk about it, an environmental influence. We found that aldehydes, which are present everywhere in our environment and are also products of many metabolic pathways in cells, can actually affect the stability and folding of BRCA2, causing inactivation of folding in a way that inactivates function, causing more mutations. And because this is a field effect, an environmental effect, it may affect more than just a single cell. And therefore, we believe that the origin of cancer in certain organs in people who carry BRCA2 mutations may be a combination of the environment of those organs, metabolism producing these types of chemicals which influence BRCA2 folding and function, and the actual hit that they carry in the germline.
So it's, again, just to follow up. So is it purely like an environmental, so the amount of cancer cells is related to the environmental stimulus, or is there a signaling mechanism from a dying cell that causes other cells to get cancer?
So far, we've been able to demonstrate in vitro, in a simple model system, only the cell autonomous effect, that is, effects at the level of individual cells. We have not yet been able to show a non-cell autonomous effect in which changes in one cell affect another cell. That's not to say they don't exist, it's just that we haven't found them yet. Our system may be too simple.
On that note, thank you so much. I certainly learned a lot, and thank you for the panelists, Vinicius and Kostya, and hopefully that'll result in collaboration between the two centers. And if there are no more questions from the audience, well, I thank everybody for their time.
Thank you very much for having me and for all the questions. I look forward to further discussions.
Thank you.
Thank you.
Bye-bye.
Bye.